structure of proteins, students, right? Structure of proteins. See students, we can discuss structure of proteins under 4 headings: primary structure, secondary structure, tertiary structure and quaternary structure, students, right? So this is also very important neat point of view, students, right? Let us try to understand what exactly primary structure is, students, right? It is very simple, students. Try to understand, right, what exactly primary structure is. So primary structure means, you see, it is the primary structure is sequence of amino acids, students, right? So primary structure refers to sequence of amino acids, that is positional information of amino acids in a protein, students, right? So that is very important, you need to remember. And you see, just imagine protein as a straight line. If you imagine protein as a straight line, students, extreme left amino acid is considered as what? First amino acid, and right, last amino acid, you can say right amino acid, you can say, students, right? So if you imagine this protein as what? Straight chain. So this is, just imagine, student, this is what? First amino acid, and extreme right amino acid is called what? Last amino acid, students. How simple it is, you see, it's very simple. So what do you understand by primary structure? Primary structure refers to sequence of amino acid, students. That means positional information of amino acid. Which one is first amino acid, which one is second amino acid, which one is third, which one is fourth, which one is right, fifth, and which one is last. Right, student? Right. So that is what? Primary structure. And if you imagine protein as a straight line, so amino acid which is present at the extreme left is called first amino acid which is present at extreme right is known as what? Last amino acid, student, right? It is very simple, students. And one more thing, first amino acid is also called what? N terminal amino acid, and last amino acid is called what, students? C terminal amino acid. Why we call first amino acid as N terminal amino acid, and why do we call what? Last amino acid as C terminal amino acid, students? You need to understand that. You can observe, okay, simply this diagram. In this diagram, you see, this is what? Extreme left amino acid, students. So here amino group will be there. So therefore first amino acid is called what? N terminal amino acid, students. And last amino acid, if you see, extreme right amino acid, that is the last amino acid, if you see, their carboxylic group is free, students. So therefore right amino acid is called C terminal amino acid, and left amino acid is called N terminal amino acid, students, right? So this is what? Primary structure of the protein, students, right? I hope you understand. It is very simple, students. Now you see secondary structure of protein, students. What exactly secondary structure of protein is? See, students, here in secondary structure what happens? One or more polypeptide chains, students, right, folded in the form of helix. You can clearly see here. So this is what? One polypeptide chain I have shown, student, right? Just imagine, this is polypeptide chain, right? So this is what? Polypeptide chain, student, right? So these are what? Amino acids. Just imagine, these are amino acid, students, right? So this is polypeptide chain. If this polypeptide chain folded in the form of helix, just imagine like this. If this polypeptide chain is folded in the form of what? Helix, right? So this kind of structure is called what? Secondary structure, students. It is very simple. Secondary structure refers to what? So sometimes what happens, students, one polypeptide chain or more than one polypeptide chain, they are folded in the form of what? Helix, student, right? You need to understand. And this kind of structure is called what? Secondary structure. And another most important thing you need to remember, in secondary structure, only some portion of the protein is folded, student. You can clearly see here. Just imagine, here one amino acid, here one amino acid, here one amino acid is there, right? Just imagine, student, right, all these things. So you see now, only some portion of the polypeptide is folded or not? Only some portion of the polypeptide is folded, student, right? So rest of the polypeptide is what? Again straight. Rest of the polypeptide is again straight chain, student, right? So that is what I have mentioned. Only some portion of the protein thread are arranged in the form of what? Helix, student, right? It's very important, you need to remember. Helix is singular and helices are plural, students. In secondary structure, generally you can find what? In proteins, not only in secondary structure, but generally in proteins, what happens, students? You can find right-handed helices. This is very important, students. Proteins generally show what? Right hand helices. Helices is plural and helix is singular, students. This is very important, you need to remember. And another important thing, students, generally in proteins, what you can see, what kind of helices you can see students, generally in proteins you can see what, right-handed helices students, right, right-handed helices as I have shown over here you can see. Now you see there are some examples, right student, right, as far as primary structure is concerned students, keratin, right. So keratin is one example. You know our hair contain a protein student, and that protein is called what, keratin. Keratin is an example for what, secondary structure. And fibroin is there in silk. You can find what student, one more protein, and that protein is what, fibroin student, right. Fibroin you can find in silk, silk fiber student, right. So fibroin is one example, and another one example is what, keratin students. Now you see students, let us try to understand how keratin, what is the structure, exact structure of what, keratin. So this is what keratin student, you can clearly see, right. So this is keratin protein, and you see students here I have shown what, fibroin, right. So this is what, fibroin student, right, just extra information this is. Now you see students, let us try to understand in secondary structure we can find what students, again you can find what, some different kinds of structure students, right. In secondary structure, secondary protein structure is of different types students, 3 different types you can say directly. One is alpha helix, one is alpha helix students. In alpha helix what happens, polypeptide gets helical shape. So you can clearly see student, polypeptide chain gets what, when polypeptide chain gets helical shape, that is called what, alpha helix. It's very simple students. Now you see beta plated students, another important second type in secondary structure is what, beta plated sheet student. What is exactly beta plated sheet? When 2 or more polypeptide chains students, you can see here, when 2 or more polypeptide chains, they are held together by intermolecular hydrogen bonds students, right. What do you mean by intermolecular hydrogen bonds? Intermolecular hydrogen bond means hydrogen bonds will be formed, right, in between amino acids of what, polypeptide chain students, right. Sometimes what happens, you see, right, one or more than one polypeptide chains students, they are held together by means of what, intermolecular hydrogen bonds, right. So this kind of structure is known as what, beta plated sheet students, right. It is very, very simple. In alpha helix, polypeptide gets, right, helical shape, alpha helix. In beta plated what happens students, right, in beta plated 2 or more than 2 polypeptide chains are held in position by intermolecular hydrogen bonds, right, and this kind of structure is known as what, beta plated sheet students. This is very important, you need to remember. And as far as collagen helix is concerned students, here what happens, you see, polypeptide, right, coil of collagen helix is further strengthened by what, establishing hydrogen bond students, and locking effect also occurs with the help of what, some additional what you can say, amino acids such as proline and hydroxyproline student, proline. So hydroxyproline and proline, right, so they provide what, locking effect students. Again hydrogen bonds will be there. It's very simple. So with this we have finished what, collagen helix. What is collagen helix? The polypeptide coil of collagen helix is strengthened. You can clearly see here, this is collagen helix students, right. So polypeptide of collagen helix is further strengthened by what, hydrogen bonds, and apart from hydrogen bonds, right, so you can find what, locking effect student, and that locking effect is because of presence of what, this proline and hydroxyproline amino acid students. So with this we have finished what, secondary structure in detail we have finished students, right. Now let us try to understand tertiary structure of the protein students. What exactly tertiary structure of the protein is? See, in tertiary structure what happens, the long polypeptide chain students, or you can say protein chain, the long protein chain or polypeptide chain, right, folded upon itself like a hollow woolen ball student, right, and giving rise to what, tertiary structure. You see here, simply I would like to show, just imagine students, so this is what, how tertiary structure is formed. You see, this is what, polypeptide chain students. This polypeptide chain, this is N terminal amino acid, first amino acid, and this is C terminal amino acid student, right. So this polypeptide chain folded upon itself in the form of hollow woolen ball, and and thus forms what, tertiary structure of the protein students. So this is very, very important students. Now you see, tertiary structure gives what, 3-dimensional view of the protein students, 3-dimensional view of the protein. And tertiary structures are absolutely essential for what, biological activities students, biological activities of proteins. Tertiary structures are absolutely necessary for biological activities of protein students, right. So enzymes will be having what, tertiary structure, right, students, including that what, substrate binding site, student, right. So here examples I have mentioned. Myoglobin is an example for what? Tertiary protein, students, and enzymes, right. So enzymes, myoglobin and enzymes, these are examples of what? Tertiary protein, students. I hope you understand. It's very simple, students. So fourth one, right. So quaternary structure. What exactly quaternary structure is? You see here, sometimes what happens, students, some proteins are an assembly of more than one polypeptide chain, students, right. So what happens generally, some proteins are formed of only one polypeptide chain, students, right. But in quaternary structure, always what happens, right. So proteins will be always having what? More than one polypeptide chain, students. So some proteins are what? An assembly. Assembly means group of what? More than one polypeptide chain, students, and those polypeptide chains are also called what? Subunits. It is very simple. Now you see the manner in which these individually folded what? Polypeptide, students, are subunits. You can say how these are arranged with respect to one another, right. Means whether they are arranged in what? Whether they are arranged in the form of linear springs of spheres, or they are arranged what? Spheres means spheres are formed of what? Polypeptides. Sphere is nothing but what? Polypeptide. Polypeptide is arranged in the form of what? Sphere, student, right. If these spheres are arranged in the form of string, it's very simple. Linear strings of what? Sphere, students, and sphere is nothing but what? Polypeptide, are spheres arranged one upon other, right, in the form of what? Cube or plate, student, right. That is the architecture, that is the structure of what? Protein, and that kind of structure is known as what, student? Quaternary structure. How simple it is, you see. Just imagine. So if you observe this figure, student, you can understand what exactly quaternary structure is. Just imagine this is what? Hemoglobin, students. So this is what? Hemoglobin. You need to understand this hemoglobin is made up of what? You see, hemoglobin consists of, students, you see, most important, hemoglobin consists of 4 polypeptide chains, right. So hemoglobin consists of 4 polypeptide chains. Polypeptide chains are nothing but what? 4 subunits, students, right. 1 2 3 4, you can see. And you know, each polypeptide chain is folded and it forms what? Sphere, student, and these spheres are arranged one above other, like in the form of what? Cube. And this kind of structure is nothing but what? Quaternary structure, students, right. So with this we have finished what? Quaternary structure as well, students, right.